Okay, so now we move to the next topic, translation or protein synthesis. Okay, and the regulation and related biotechnological tools. Okay, so we have learned earlier that like no, sorry, in this course I didn't. So I would like to start this chapter with a little bit of history. Okay, so you think back and you will realize that DNA as the so like Mendel showed that traits are like heritable, right? But it was not clear what is that biological material that is responsible for inheritance, right? And you remember all those experiments, perhaps, or Oswald experiments, and uh, all the experiments that eventually led to the discovery of DNA as the genetic material and the double helix structure, etc., etc. Right. So while all this work was going on, there was another question that was bothering the scientific community. That is, how is because you need to understand that early 19th century biology research was heavily dominated by the chemists, biochemists. They understood a whole lot about proteins and enzymes, and they pretty much established that it is the proteins who actually do all the job. So therefore, if DNA is the heritable material, then there has to be a relationship between the DNA and the proteins made. Okay? The first such data that was out there was from an experiment by Garrod in 1902. So, like not experiment, observation. So, there is a disease called alkaptonuria. In alkaptonuria, there is a defect in the phenylalanine metabolism, and therefore the urine turned black. And this disorder is inherited following Mendelian ratio. Therefore, uh, this is the first evidence. Once everybody knew that DNA is the basis of heredity, then it was clear. Like Garrod's experiments were reported in 1902, but later on people could make sense that okay, so then there is some relationship between inheritance and DNA, and in the case of alkaptonuria, inheritance and enzymes. The enzymes were gods in those days. So therefore, there is a relationship between DNA and protein, because enzymes are proteins. Okay. Another experimental evidence came from little unlikely sources. That is, there is this single cell plant called Acetabularia, and this Acetabularia cells are composed of three segments: the foot or the base, where the nucleus is, the stalk, and the cap. Okay, and this cap, the appearance varied between species. So there were two different species of Acetabularia. One, the Mediterranean, has a smooth and disc-shaped cap, while Crenulata has a branched flower-like cap. And when Hammerling, he exchanged the feet of two different species of Acetabularia, he realized that soon after the cap morphology changed. Okay, the cap morphology became the one like the one which food has been provided. Okay, so therefore there is a relationship between, and if you artificially put in an intact acetabularia, if you put artificially another nucleus, then the cap assumes an intermediary structure, a hybrid structure. Therefore, there was relationship that the nucleus can actually influence the morphology, and morphology is directly related to protein. So these were two lines of evidences that said that there is a relationship between DNA and protein. But the question is that how is that relationship? Is this a direct relationship? Is DNA itself dictating which kinds of proteins to be made, or is there something else? By that time, RNA as a new chemical entity was discovered. It was known to be metastable, and people knew that there are ribosomal RNA, tRNA, ribosomes have RNA. So, in fact, there were theories proposed by very senior established researchers that they thought that it is the RNA that dictates which protein will be made, and it is the ribosome that dictates which protein will be made. In fact, they thought that the RNA present in the ribosome, like each ribosome is programmed to make one kind of protein only. Okay. And there are many other theories. Today we might think and we might think that those were crazy theories, but you need to transport yourself back to 1940s and 50s when or even 60s, early not 60s, but certainly 50s, when people really had no idea about molecular genetics. This is an era when the DNA was not really known, double helix structure was not discovered, sequencing was not, DNA sequencing was not really there. So it was a very different era. So therefore, the next set of experiments were designed to test Sidney Brenner, Francois Jacob, and Matt Michelson, they, this is fantastic scientific collaboration, by the way. It is cross-continental, absolute stalwarts of science. They thought that they want to, look into this problem at greater depth. And if you read this particular paper, you'll see that they are actually talking about all the three different hypotheses. And then they are trying to say that, okay, let us presume that RNA is the intermediary between DNA and protein. And somehow, RNA is decoded by the ribosomes to make proteins. Okay? So how did they design those experiments? And you just see the genius. They knew that upon bacteriophage infection, and in this particular case, they took bacteriophage T2, the DNA synthesis of the host bacteria stops. Okay? So bacterial DNA is no longer synthesized upon bacteriophage infection. And soon thereafter, within 10 minutes, lot of low stability RNAs are made. Now we know that those are the mRNAs. In those days, that was the terminology used. And those RNA had composition, not sequence, okay? Composition very similar to that of the phage genome, okay? And then, 60% of the protein that were generated after infection, they coded for, they coded for bacteriophage coat proteins. Therefore, phage DNA that is going inside the cell is dictating, or at least inducing, production of RNA, which has composition very similar to phage DNA. And soon after results, it triggers production of proteins that are typical of bacteriophage. Okay, so if the phage RNA, so if there had to be, pre-programmed ribosomes, then for making bacteriophage proteins, phage will have to program ribosomes, right? So you see how they cracked this problem. So what they did is they grew bacteria in the presence of tritium. Okay, like they used to call those heavy medium, just because like tritium has molecular weight of 3H3. Right? So and then like if you grow the bacteria in presence of tritium, then everything that has an hydrogen atom will be radioactive, right, inside the bacteria, including the ribosomes. Then they infected these bacteria. So therefore, if the ribosome is radioactive, then you know that this ribosome was produced even before phage infection happened, right? Now you infect it with phage, and then you switch the bacteria to cold medium. It is no longer tritiated medium, okay? So now after phage infection, therefore, if phage programs ribosomes, then phage programmed ribosomes cannot be radioactive, right? And then you add just inorganic phosphate, radio-labeled inorganic phosphate. So therefore, everything that will incorporate phosphate, ATP, GTP, UTP, whatever, and phosphorylated proteins, everything will be radioactive, including the RNA. And remember, right after phage infection, within 10 minutes or so, you make lot of phage-specific RNA, right? Now you simply ask the question that these radioactive RNA, which will be primarily from phage, right? Because that's what it is. Now these radioactive RNA, are they with heavy ribosome or with normal light ribosome? Light ribosome will be produced only after phage infection, because that's when they switched it to the light medium. And to cut a long story short, when they ran it on the density gradient, I think they used cesium chloride gradient in those days, a density gradient, they could see that the radioactive ribosome and P32 level RNA, they are peaking in the same tube. That means that these ribosomes, see, like RNA has much lighter molecular weight than ribosome, right? So normally they should not come in the same tube. The fact that they are coming in the same tube, meaning these RNA molecules are associated with the ribosome, and they are associated with the heavy ribosome. Therefore, for production of phage proteins, even then the ribosomes that were produced before infection, only they are being used. But the phage RNA are being loaded onto them. And then that phage proteins are made. So therefore, that makes it a very solid argument that the ribosomes are not specialized. It is like which RNA they are associated with will dictate which protein will be made, right? And like then by this time electron microscopy has improved quite significantly, and they could actually see that on a growing RNA transcript, there are ribosomes, not one but two or many ribosomes, which they called polyribosomes. So they knew that ribosomes actually crawl over the RNA. So everything taken together, it was clear that RNA is being decoded by ribosome. Now the question comes that what kind of code is it? Okay, is it overlapping code, non-overlapping code? Will there be comma in between, or it is a contiguous code? Lots of different questions were there. Because see, you try to understand that we are talking about an era where people were discovering, like the cracking the genetic code, perhaps is the biggest thing that happened in the 19th century in biology, right? So and there is no set rule, because they are discovering the rule for the first time. So anything was on the cards. In fact, soon after DNA double helix structure was discovered, and Watson and Crick talked about the possible mechanism by which it can be replicated, and the genetic code was being talked about. So Gamow in Russia, he formed a club called the RNA Tie Club. Okay, they took it upon themselves to crack the genetic code. Okay, and the RNA Tie Club had 20 members, who is who of molecular biological science in those days, and each one were given one amino acid. Okay, like a 20-member RNA Tie Club, each one had a tie with one amino acid associated. So the team would crack the genetic code, and these are all established scientists. And my hypothesis has always been that new discoveries are seldom made by established people. New discoveries are made by people who are absolutely fresh. They don't have any baggage of thinking in a particular manner, and they just make discoveries. All you need to have is resolve and determination, and you can do it. Okay? So Crick and Sidney Brenner. Sidney Brenner is perhaps one of the smartest, brightest scientists that biological science has ever seen. So they, did experiments and showed that if you delete a base, add a base in an RNA, then this kind of now we know that those are friendship mutations. They significantly reduced the infectivity of T7 bacteriophages towards the bacteria. Therefore, this particular code is very, very important. Now, as always, there will be some very, apparently innocuous discoveries that made things possible. There was an enzyme that was characterized. It's called polynucleotide phosphorylase. That could make RNA in a non-template dependent manner by simply using dNTP, the RNA version of NTP, like a NDP, the dinucleotide, the ADP, UDP type. You take multiple, like N RNDPs, you use this enzyme, and it can form RNMP-N, meaning polymer of RNMPs, and inorganic phosphate is released. So now, see, what was the problem of dealing with DNA template derived RNA that people could make, right? What is the problem? There you cannot dictate what the sequence of the RNA will be. And this is an era when, the DNA synthesis, in vitro DNA synthesis, etc., was still not there, right? So the scientists needed a tool to make RNA of their choice, because then and only then they can ask the questions that if this is the RNA, what kind of protein it will make. So this enzyme characterization was a huge tool for them. And then, entered a guy called Marshall Nirenbach. Marshall Nirenbach was farthest from being a molecular geneticist. He was doing postdoc in NIH. His PhD work was uptake of sugar in cancer cells. So it was basically a physiology guy. But he said, I really want to know more about protein synthesis. And if I want to know more about protein synthesis, I need to know about the genetic code. Okay, but he had no formal training. So how did he approach? So what he argued, that if I can make a bacterial extract which will not have any RNA of its own, and then I will supply RNA that I know the sequence, and then I want to see if it codes for a particular kind of amino acid polymerization. So each tube, so he made 20 different tubes. Each tube will have 19 amino acids cold and only one amino acid radioactive. And through months of hard work, Nirenbach and his postdoctoral colleague Mathai, they set up this whole system. And if you read the history, you will find it on NIH website. It's their point of pride, because Nirenbach was the first intramural NIH scientist to have received a Nobel Prize. So they display a lot of things about Nirenbach's experiment. So at 3 a.m. one day, Mathai figured out that the tube that had radioactive leucine has polymerized. As I told you earlier, the way the polymerization would be done, would be diagnosed is like this. So, you make these short RNAs with known sequence, and you add a mixture of ribosomes, tRNA, but the whole cytosolic extract without RNA. Okay. And then you mix them, and because this is radioactive, so whatever amino acid it is supposed to polymerize, it will polymerize, and then you pour it through a filter paper. Now, monomeric radio-labeled amino acid will pass through the filter paper, but if it is polymeric, then it will not pass through the filter paper. Okay, so that's how they would determine it. And this particular setup is slightly different. The slide is in wrong order, but the basis is the same. But it should go a little later. But that's okay. The basis is the same. So this is the most famous PolyU experiment. So Marshall Nirenbach, therefore, using an artificial RNA with known sequence, homopolymer of U, would show that this directs polymerization of phenylalanine. So he knew that a stretch of U's can direct polymerization of so U was likely the code for phenylalanine. It was kind of guessed that it will be 3-letter code, because 2-letter will give only 16 amino acids possible, and in reality there are 20 different amino acids, and 4 letters will mean like 256 different kinds of probabilities, and while you only have 64 20 different amino acids, so you really don't need it. So it was guessed that it will be 3-letter code. And then, subsequently he made poly A, poly C, and he could establish the first set of rules, that UUU codes for phenylalanine, AAA codes for lysine, CCC codes for proline. Okay, so that's he knew. Then he did something differently, that he made a polymer of alternating A's and C's. Okay, so if you are alternating A's and C's, you will make 2 different kinds of codons only, ACA or CAC. Okay, then you do the same experiment, and you see that it makes a polypeptide which has alternatively threonine or histidine, threonine and histidine, threonine and histidine. But this experiment cannot tell you whether ACA codes for threonine or ACA codes for histidine, and vice versa, right? So to crack that, this is exactly like solving a puzzle. Then what he did is he made AAC triplet and polymerized them. Okay, so they are like AAC, AAC, AAC. Now you can read it in 3 different frames, right? AAC like this, or ACA, ACA like this, or CAA, CAA, CAA like this, right? And then he did the experiment again, and he figured out that he gets asparagine or threonine or glutamine. Now, between this experiment and this experiment, the only codon, possible codon that is common between the two is ACA. Therefore, threonine was assigned the codon ACA, and so on and so forth. Eventually, all the 64 codons were, discovered by Nirenbach, and in parallel. So another defining experiment of Nirenbach was just give me a minute. So this slide should be right before this slide. No? That's it. Another defining experiment of Nirenbach was done in collaboration with his graduate student, Phil Leader. And I should tell you my point of pride that when I was at Harvard Medical School in the genetics department, Phil Leader was the chairperson of the department. And on our department, the genetic code was displayed on the department wall. It was an enormous matter of pride. So what Phil Leader did is the same experiment. These 20 different extracts and the ribosome-free bacterial extracts that had all the possible tRNAs with the amino acid attached. Okay, these are called charged tRNAs, and in each a different amino acid was radio labeled. Okay, and the only thing that he did is that he added triplet codons and showed that if you add this triplet codon, then the radioactivity is arrested on the filter paper. And this arresting the radioactivity on the filter paper is dependent on presence of ribosomes. You see, when you don't have ribosome, then the tRNA, even the charged tRNA, is small enough molecule to pass through the filter paper. But if you have the ribosome, then it is arrested in the ribosome-bound tRNA codon complex. Okay? Then it is retained over the filter paper. And this way, they could show, all the different triplet codons they made, and they could figure out exactly which codon is coding for which amino acid. Okay? Hargobind Khurana, in parallel, was running the same set of experiments. And his forte was that he could really make all sorts of RNA polymers very well. He was a very well-trained chemist. So, he also in parallel discovered, the codons. Okay. So, together, then Nirenbach and Khurana really cracked the genetic code. Now we all know what the genetic code is. And, we also know that some of the amino acids are coded by multiple different codons and all those things we know. So in 1968, Nirenbach and Khurana, they received their Nobel Prize along with Robert Hawley. Robert Hawley did not really crack the genetic code, but he deciphered the tRNA structure, including all the chemical modifications of tRNA, etc. Enormous amount of work. So whatever we know about tRNA today is largely due to Robert Hawley's work. Okay, so they became overnight celebrities. So what do we know about the genetic code today? That it is made up of codons, triplet codons. The codons do not overlap. That is, this particular sequence, it contains two different codons, and there is no comma. The code includes punctuation in the form of 3 stop codons, the UAA, UAG, UGA. These are the 3 stop codons. So they don't really code for an amino acid, but if the ribosome hits them, it signals stop protein synthesis. Okay. The genetic code is called degenerate because some of the amino acids are coded by multiple different codons, so there is degeneracy. For any protein to be made, like from DNA to RNA to protein, it always has to have a fixed start site. And if it starts at the fixed start site, it will only, because it's a comma-less genetic code, so it will only be able to read in multiples of 3 till it hits one of these punctuation marks and falls off. Okay. And it is read always from 5' to 3' end. And if there are mutations or errors in the DNA, the message may be changed. The start codon is almost always AUG. There are some exceptions. In some bacteria, it can be GUG, and rarely it is CUG as well. And if you have an RNA sequence, then it can potentially be read in three different frames, but because of the AUG, the reading frame is fixed. And even if there is a possibility, it will not be able to make the amino acids that will be, or the amino acid polymer that will be possible with an alternative reading frame, because this dictates and sets the reading frame. If for some reason the frame shifts, sometimes it happens that the ribosome skips a base, or sometimes it reads four nucleotides as one codon, then the frame will shift, and that will have deleterious effects. But fortunately, it is extremely, extremely rare. Like few dozen examples are no, not more than that. And the genetic code is universal. No matter which organism it is the same codon that works, and that is a very, very strong argument in favor of the fact that life on Earth evolved once and only once. Okay, but the genetic code does vary a little bit in many mitochondria, and some protozoans and Acetabularia, etc., etc. And most of these changes involve reading of normal stop codons as amino acid. This is not an exchange of one amino acid for another. It's just that like the UAA or UGA, etc., the codes for amino acid in this case. But these exceptions to the code to most likely later evolutionary development. Okay, so today in this first lecture, let us stop right here, and in the next lecture we will get into the RNA, different types of RNA and protein synthesis process. Okay, um, thank you. We'll see you soon. Okay.